sample, say soil, and how do we make sure that like you are able to extract all the DNA from the soil sample? Is it possible to extract all the DNA from soil sample? No, depending on the matrix which you select, it becomes really difficult. If you have pure culture from a broth or a culture medium, maybe it is possible to extract all the DNA from the bacterial cells. But again, like it's a theoretically it may look like possible, but even with the culture, you won't be able to extract all the DNA. More so in the case of soil sample or a matrix which is having different other organisms, having complex ecosystem samples. Okay, in that case, well, you need to be really careful in handling the soil for extracting DNA or any sample for extracting DNA. I might cite like how do we quantify the extraction efficiency of DNA from a complex matrix? What is the way you think that, okay, what is my extraction efficiency? How do you how do you set an experiment, or how will you quantify the extraction efficiency of extracting DNA from soil sample? Can you think of a way? Just imagine, sir. No, theoretically we want to extract all the DNA, all the bacterial DNA or microbial DNA from soil sample. But you are not sure whether you are able to extract all the DNA. I am asking you, what is the way you can think of now to quantify the extraction efficiency? The extraction for DNA, what is the efficiency? How will you quantify it? Simple way is
sample to chemical ratio.
Sample to chemical ratio. It's not a chemical reaction to, No, okay, let us not get into too much of discussion. One of the ways is introduce a reporter gene, okay, from using a bacteria. You select a bacterium with a new gene which is normally not present in the sample. Then if you know the titer value of the bacteria, then you can know what is the after you do the extraction, you will be able to quantify the extraction efficiency. There are ways of quantifying the extraction efficiency of DNA. But as I told you in the class, before you go for the extraction, you actually quantify the sample which you are taking for extraction. So in my lab, in our laboratory, what we normally do is we use kits for extracting DNA. Of course, you can also use other methods, but if you use kit, like it's easy and you are having a dependable or reliable DNA getting extracted from the sample. So after you extract them, you again use another kit to purify the inhibitors, because in soil you have so many inhibitors, like you have polyphenols, humic acid, and so many stuff which are present in it. They will inhibit the PCR reaction. So you take the DNA after extracting the DNA from your sample, then use the purification kit, then purify them, or you run a PCR reaction, find out whether you have any inhibition or of the reaction, then you go for the other real-time analysis. Okay, in this today's class, I think Ilekya would give you the DNA sample which has been quantified using nanodrop. So we know what is the amount of DNA which is present in that sample, and then you also will be given primers, 2 set of primers. Well, you can count, those you use the standard protocols, and then calculate the amount of primers to be used in the final reaction. Normally we said 20 microliter reaction, so we quantify the primers which are required for that. And then we also use cyber grain master mix, again to make sure that like our reaction is uniform in all the cases. And otherwise, sometimes you may use, most of the mistakes come from pipetting error. If you do not do it properly, so you are, all the reaction, DNTPs, enzymes, water, might create problem when it comes to amplification efficiency. Try to minimize the number of times you pipette any of the reagents and prepare the PCR curve. And we had several difficulties in the beginning, because most of the reactions, we were not, some of them were failing. In some cases, even NTC, no template control, were also showing amplification. So finally, we have to design our own PCR cabinet, which is sterile one, and we use only in that. When I asked Alekia, where do you do it? She said we do it on the open top bench table itself. So now we are not doing so many replication because open remains like sometimes we have problem in quantification because your no template control will also show amplification. So as far as possible, you try to keep the area clean. In case you don't have a PCR cabinet, keep the area clean by wiping it with ethanol or disinfectant. And so keep the area clean because we need not calculate the false positives and try to report those results. So you need to keep the whole area clean. And well, as in this lab, you prepare all your reagents, prepare the PCR, real-time PCR reagent, whatever you want to do it in a place which is other than where you have an equipment. Keep that PCR machine, real-time PCR equipment should be in a well-ventilated place, and and also make sure that like it is in a say temperature range of 20 to 27, 25, not more than that, because otherwise your the sensor will have problems. So all those things you have to take care about. So today you will be given 5 groups will be given DNA template and primers and cyber grain master mix and also nucleus free water. You just prepare 20 microliter for PCR reaction, and she will show you how to set the program. So each equipment has got a way of doing it. So they have in Roche machine has got a monitor where you will be able to do everything by touch screen, and also you can use a laptop which is dedicated to the machine itself and do the analysis there itself. Maybe if after everything is over, after about 1.5 hours of PCR reaction, she will show you how to note down the CQ value from the software itself, and that's also will be done in this practical. You have anything to ask? Okay, so she will just show how to prepare it, or she will give the materials to one of you.
So individually they will be adding everything?
5 groups, one person. Individually you can ask them to add. Okay, this is only to see how the pipetting error is going to change your values, because the same DNA is being given, same primer set is going to be given, cyber master mix is also same, and then we can see like whether our standard deviation values is very low or we are actually having problem with pipetting. That could be seen. But otherwise, when you start an experiment, you make sure that like your DNA is of high quality and you are not committing so many errors in the pipetting, and reaction cups is well set before you take them to the real-time machine. Okay, okay. And she is telling me that she has brought a different DNA sample. So in addition to pipetting, which we may introduce some variations, we will also see like how DNA samples of 5 different environmental matrices, what is the gene which copies which we are going to quantify them. Okay.
So like previous classes, any one from one batch can come to handle.
Maybe one of you come and do it, and then the next one will come. I mean, one after another one can come.
After another one team was coming.
So you note down the number and order, so that you know which environmental sample you are going to use for each group. Okay. Okay, for others, for those who wanted to do, let them do. I'll just discuss few points of using absolute quantitation in case of microbiology. You know, we all been doing what do you call culture-based quantification of microorganism from several matrices. So what we do is we will take the sample, we will dilute them, we will plate it out on a media of our interest, then quantify them. How many days normally takes for you to quantify bacterial culture from an environmental sample? Just say. So it depends on the organism of interest. Some organism will come up within 24 hours, some will come several days. So if you are interested in quantifying, say, methanogens or organisms like chemoautotrophic organisms, how many days you anticipate you will be working for quantification? How many days of incubation you require for quantification? How long? How long? Some days. You just give me an estimate. 3 months, 2 months. If you go for anaerobic organism, it takes several months, 3 months, 2 months. And then, you know, to prepare the medium, how many reagents or how many chemicals you may require to prepare. Some organisms, those which are, you know, very what you call copious organisms, they may require less number of chemicals, so you will be able to grow them. Some of them are called as fastidious organisms. They require a long list of chemicals. Some may even run into 20 to 30 chemicals to prepare a culture, to grow the cultures. But if you are able to extract DNA from an environmental sample, and if you know certain markers, environmental gene markers, and which are specific for a particular group of organism, will it save time for you? So it is easy to quantify. Okay, suppose if you are given a sample and you want to quantify, say, about 3 or 4 different genera from the same sample, how many samples you may have to handle? 5, 6 genera you wanted to quantify from a particular sample. So you need to dilute, prepare a different, at least 6, 7 media. 6 means 6 media you have to prepare them and incubate them in a particular condition, maybe high temperature, low temperature, with oxygen, without oxygen, with light, without light. So you need to have environmental incubation condition also different for certain group of organism. So you are going to spend more time, more amount of efforts to quantify simultaneously different group of organism from a single sample. But if you are using molecular methods, from one sample, if you are able to extract DNA and you have sufficient quantity of DNA in your hands, how many you are able to quantify? As many as you could. If you have markers which are very specific, I am saying about bacterial markers, you should have. 16S may not be an interesting marker for us because several organisms, they have multiple copies of 16S gene. So you are not able to relate the gene with the number of microorganisms present in the sample. But if you look for certain, you know, genes like ammonium oxidizing bacterial genes, or you know, you have several groups, NIRS of denitrification. So those genes are very specific for one microbial guild, or what you call as a physiological group. Then it is easy to relate like what is the abundance of a particular group of organism in an environmental sample. So these kind of reliable method or quick methods are possible using molecular methods. So that's one of the reasons like we wanted to move on to the genomic based molecular methods for several investigations. So if you go for culture based methods, you are going to wait a long period to quantify them, and some of the methods like NPN counting, which are also being done in several laboratories. In fact, if you leave your NPN tubes for a longer period, all of them will give the same numbers. So they will reach the maximum number which is possible for an NPN tube to, you can estimate. So we have certain difficulties with the culture based methods. Those can be overcome using molecular methods. That's the reason. And also if you want to actually monitor the temporal variation in a particular group of organism, how many samples you may have to handle. Suppose if you want to know what is the population of a NIF, a group of organism on 10th day, and what is the population on 20th day, on 30th day and 40th day. So if you want to see the temporal variations, how many times you have to quantify them? So it takes a lot of efforts for you to monitor those changes. If you wanted to see the changes within the space, like different depth level, the surface 5 centimeter, 10 centimeter. So again you have to sample them carefully, take them out of those samples, prepare dilution, and then plate them out, and then count the number of colonies, say about the population which is present in those areas. But now using these methods, your sample preparation will also get simpler, easier, and you will be able to get a good amount of DNA to monitor those changes on a temporal basis on a spatial scale. And if you combine this data with metagenomic data, you have a lot of advantages. So if you know what are the population of what are different group of microorganisms present in a sample, in an environmental matrix, then you will be able to follow them on a temporal scale. You can see the dynamic changes over a period of time. You can see the changes within a different spatial scales. All those things are possible. So once you standardize the method in the sense you identify a gene of interest which is unique to a particular group of organisms, then it is easy for you to follow them up using molecular methods. How many groups have done the preparation now? Only one? No one. Okay, the other issue I want you to learn is, and what is the cost of a real-time PCR machine? Huh? 8 to 9 lakhs. What is the cost of a normal PCR machine? 3 to 4 lakhs. So you be aware about this cost factor, because when you say 8 to 9 lakhs, may have certain features which may not be, you may not be able to extend certain other functions and all. So the equipment, the brand should also, the way you select will actually give you some advantages over choosing different methods. A PCR without hard start function, PCR without a gradient function machine will be a little handicap for us. So understand what do you want to select is also important, right? And don't think that real-time PCR is the new development. We have some other additions which have come up in the field, which includes droplet digital PCR. Well, we have not talked about, we have not discussed about it in the class. That is another version, or rather advanced version of a real-time quantification of DNA or PCR amplicon using droplet digital PCR, which actually improves the reliability and accuracy of your quantitation. So which will cost about, right now it costs about 60 lakhs. So like your PCR machine, when the cost comes down, we will be able to see many of those machines in several laboratories. Okay.
So totally 6 reactions we will do. I'm preparing a 7 reaction for error correction. Now I'm preparing master mix, nucleus free water, forward primer, reverse primer, VSA, except DNA, and DNA will be added finally to the strips.
We have two formats or types of PCR cups for Roche machine. So one is known as multi-well plate, which is 96 well plate. The other one is PCR strips, 8 well PCR strips. For this class purpose, we are using only 8 well strip. She has already prepared the master mix with the primers and also with the template. Without template. Template will be given to each one of you, and you will have to add the template. Template is about 6 microliter?
4 microliter.
4 microliter. When you prepare all these reagents, you always have 10% more of it, so that your handling time, when you handle with the pipette, there could be some error, so you will run short of chemical in the last cup, which you can avoid by having more volume in the preparation. I wish we do this whole preparation in a cabinet, which is free from contamination. Always do it. Either you can buy it, which will be about 1.2 to 2 lakh rupee PCR cabinet, or you make one by yourself. You can have a Perpex glass and plate and make a cabinet and put a UV lamp in, try to keep the area clean and try to use it. Also, your pipette should be always either sterilized or autoclaved and roll it up in aluminum foil and reserve a set of pipette only for real autoclaved. Do not use the normal pipette for real time. And although these operations are very simple, but if you don't take enough precautions, you may end up in having false positive results, which will frustrate you. So avoid those minor mistakes.
Only one file.
You guys had
You meet him. You go and meet there. You have to write letter. Or else he will not upload more.
Yeah, I met him. I was an
He
application.
was saying you have to go to his room and only write letter.
Now,
No, if possible.
Presentation for what? Someone saying that you're going for
PhD में लाइव चल
कोई प्रोग्राम हमारा असर नहीं था, कुछ लोगों को 8 हज़ार अभी PhD में बढ़ने तो अभी तो PhD में 14 या 15 तो वो बढ़कर 24 तो 2-3 साल बाद PhD में 25 साल और अभी तो MS सारे लोग हैं ना दुनिया मोस्ट है ज़्यादा छोटा और ये 2 या 3 बाकी सब मेरे सीनियर कोई 1 साल का, कोई 2 साल का। हाँ खाने का नहीं वेस्ट बंगाल से। आपका घर किधर? बिहार से तो है, बहुत लोग हैं बिहार से। वो बिहार से, लेकिन बिहार में ज़्यादा रहा नहीं। वो बिहार से घर है उसका, लेकिन जो है उत्तराखंड में रहते थे, बेंगलुरु में रहते थे। बंगाल उत्तराखंड। आप इधर कैसे? हमारे मेस में था, लेकिन वो चला गया। जानते हो शिशु हॉस्टल में एक लड़का था, खाने पीने घूमने
उसी टाइम चाय ब्रेक छोड़ देंगे, ठीक है? उधर शूट करके आके आप लोड करके हम चाय ब्रेक
किधर करना है?
बंद
करो। स्टार्ट का बटन है क्या वो?
वो हो गया
स्टार्ट हो गया। To the interview program or already we are interested? Go enter. No, this is some Asarani leather disease.
No, actually the template we used for that program, and there we had
The same template you change.
same template we used to
Change the parameter.
but the date will be highlighting so we can make it out which program.
रिकॉर्ड हो रहा है यही, वहाँ पे जाएगा साउंड। आप लोग
हाँ,
बात कर
डिस्कस ही कर रहे हैं ना हम।
हाँ अच्छा, उसके बारे में कर रहे हैं।
उसके बारे में हम डिस्कस कर रहे हैं।
हाँ वो पहली बार में
हो गया?
हो गया।
Program you have to explain ना।
அது வந்துட்டு ஒன்ஸ் டேட்டா வந்தோன தான் சார்
ओके 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 थोड़ी देर वेट करिए। हाँ
इसको भी
Okay. While setting up in that machine also, no, you please always write it down. Otherwise, if you will put the at different position your sample, then you will not get any type of nanopicture. That is what I am saying, that since we have taken the picture, now we are confirmed, otherwise we could have messed up, right? इसमें शायद सॉफ्टवेयर का कुछ इशू आ रहा है, मल्टीपल टाइम्स नहीं खुल रहा है, थोड़ा टाइम, तो इसको एक बार कर लेंगे, इसको शायद एंटीवायरस नहीं होगा। चलो मैं इसको निकाल सकती हूँ, ये तुम लोगों को चाहिए क्या निकाल सकते हैं, ये लो, ये तुम अपने पास लो, ये पेन ड्राइव है, वहाँ से डाटा ले लीजिए, वहाँ से डाटा ले लीजिए।
That was a dramatic transformation, converting bacteria into fungi. Amazing. My God. Just close it. Press C. Yeah. Press C. Press C. Yeah, that's okay. Okay. I hope you understood the calculation part of your PCR data, which is coming from the machine. Okay. With that, we conclude the qPCR experiments. Please recall, you have been exposed to two experiments. One is relative quantitation, that is mostly for gene expression analysis, and the second one is absolute quantitation of microorganisms or pathogens, also called as copy number estimation of the target. So, hope you understood the calculation part of both experiments. That is more important. Okay. So, I also highlighted some of the calculation formulas people have evolved. I think you must be using one of those, double delta CT method. It's the most popular one. Okay. Now we will move on to the follow-up experiment, that was transformation of bacteria for expression of green fluorescent protein coding G. Okay. The plates are here already. The groups, whoever has done it, the group leader can collect the plate and take the observation I am not sure whether you have taken the observation yet. Not yet? Not yet open, okay. Let us hope some babies are inside. Huh? So, now you open and see. Before that, I will show you what you expect out of this experiment. That's more important, okay. This is what you were exposed to the other day. You started from glycerol stock of the bacteria in question, and you have taken a single colony, and this is what is called as competent cell preparation. Yeah, what you did, 3-4 rounds of 300 millimolar sucrose washing you did. Then final you suspended in very small volume, and that was frozen in minus 80 degrees Celsius for further use. And if you want to do the transformation immediately you can proceed with the transformation. Okay. And this is a kind of a plasmids what we used. PBK mini TN7, GFP2 GM10 is the name of the vector, and this vector is very unique in the sense it get integrated into the neutral site in the bacterial genome. The neutral site is lying somewhere downstream of a gene what is called as GLMS. Okay, and this is maintained in E. coli host, that is XLI blue, and these hosts are now commercially available. So you just put up a request with the company, and you pay the necessary amount, they will send you. And this is resistant to gentamicin at 25 microgram per ml. And the helper plasmid what we used in our experiment as plasmid, that is PUX PF13 AMP100. This is a helper plasmid that guides your donor plasmid to integrate into the neutral site in the bacterial genome. And this is having ampicillin resistance at 100 microgram per ml. Okay. So normally we go for 1:1 ratio of mixing. It may be 50-50, that means 50 nanogram of DNA of your donor and 50 nanogram of DNA of your helper. You mix it, and you this mixture you mix it with your competent cell, 50 microliter of competent cell, which is having 10 to the power 9 to 10 to the power 10 cells. Okay. This is the actual electroporation, and the competent cells, of course I put here 100 to 200 microliters. The volume varies, you can it all decided by you, and you mix both and subject to the electroporation. So the conditions are already given here. The pre-chilled EQS, that's very, very important, right? Then 2.4 kilovolt, and the time constant around 4.6, which I think one of your samples we got this exactly the same reading, right? And this is the downstream work what you are supposed to do in transformation. So once you do the transformation, you can put up this slide, you will see a much better one. Yeah, you can see here. Yeah, you can put up this one also. You can see here, this is the three different plates that is showing the kind of colonies, right? And the beauty here is the colony itself is giving fluorescence, right? Are you not seeing some slight tinge of green here? The colonies, little bit of tinge, right? So that means the colonies are the protein is expressing. I would say that an excellent expression of the protein in the cells. Okay, so then you have to preserve the colonies, and you have plenty of options here. The photograph was taken from our own group. So one of the students got this many colonies. When you have a colony like this, you have an opportunity to take the best one, which is giving exceptional fluorescence. Okay, and these are all some of the factor you need to keep in your mind. Right in the colony itself, you can differentiate which one is giving excellent fluorescence and which one is giving intermediate type of fluorescence, or which one is not giving fluorescence also. So this is a good expression, this is a delayed expression, so these are all some of the observations. Now this is what you call identifying the colony right on the plate. Okay. Now actual confirmation you are supposed to do in a microscopy. The microscopic technique is a confocal laser scanning microscopy or fluorescent microscopy. Remember, not necessarily you need to have CLSM all the time. You can use a fluorescent microscopy technique also. The fluorescent microscopes, either upright or inverted microscope, I think most of the labs are having this kind of facilities. You take them on board, and this is how the colony really looks. This is one of the colonies. We just touch it and make a slide. You don't have to do anything, and take them in the fluorescent microscopy, and you start seeing something like this, right? So beautiful fluorescence, right? And very uniform, okay, very consistent fluorescence. Okay, this is how the colony looks in microscope. The cells looks in microscope. Okay, yeah, these are all the colonies in microscope. Yeah, now this is a PCR confirmation. Yeah, it's pretty weak here. So the primers are available, which is going to give you around 150 base pair amplicon. Remember, this primer, the primer sets are very unique here. One of the primer is going to complement with the bacterial genome, that is GLS-M, and another one is going to bind with the vector itself, right? So only the integrated segment is amplified. Okay, if there is no integration, you won't get any amplification. So 150 base pair amplicon if you get it, and you will get, you can say that okay, the integration is taking place. Of course, the integration is quite obvious here. Without integration, you are not going to see the fluorescence. So that is quite obvious here, but still, just for the confirmation sake, you can do this, the PCR confirmation of GFP insert. So the utility part. What is utility part? This is one slide I brought from my own paper. The paper we published in Environmental Microbiology. So basically what we did, the GFP tagged bacteria, it was inoculated in the rhizosphere. Okay, rhizosphere of tomato. Okay, so after 15 days, we started analyzing different plant segments, starting from roots, stem, and also leaf. Right, you just take the section, thin section, and you treat those tissues in 4% paraformaldehyde. Okay, then you start exposing the different tissues to microscope, either confocal microscope or fluorescent microscope. Since you are using tissue, plant tissue, it is better you use confocal microscopy. Thing is, you sometimes the deep-seated bacteria you may not be able to see in normal fluorescent microscope. You need laser scanning is needed. So if you do that, you will start seeing the bacterial colonization everywhere. Okay, this is something like this. A bacterial, I would say the group of bacteria joined together and comfortably in the leaf. That is within 15 days. So that gives you an idea. The internal migration or internal movement of bacteria right from the soil up to the leaf. You can track it. So the bacterial transformation for reporter gene expression is mostly for tracking the behavior of your bacterial cells within the plant system. So this is an experiment we have conducted under the microbes. You can prove that the organism is an endophyte or not, right? So this morning also one of the students presented the thesis. She inoculated rice, and the same experiment was conducted, and she could beautifully prove that okay, the bacterium is deep inside the rice root and rice stem. Those kind of observations can be made. This is one of the direct proof that the organism is truly endophyte. Okay, this is one of the best techniques for proving the endophytism of a bacteria. Okay, so with that, I think we'll conclude this bacterial transformation and record all those images in your mind. When you do this experiment in future, and try to look for such a colonies on your plate. Okay, now you just open the plate and see whether such colonies are appearing in your plate. Okay, I would like to see the first group who got very nice reading. So let us see whether it is there or not. Okay, by chance if you see fungi, don't think that you really did a wonderful transformation. It is a poor handling of petri plate. Okay, you should not see fungus here. Okay, if somebody is seeing fungus, and we will not give certificate. All right. Okay. Now each group you pick your plates and observe whatever you are seeing, that you record in your notebook. Okay. Okay. Yeah. Yeah. So how many of you? What is this? This is Pseudomonas. No? Why it is red in color? The media is added with a chemical called 2,3,5-triphenyl tetrasodium chloride. Okay, this is converted into a insoluble formazole, and this formazole is trapped in the bacterial cytoplasm. And this is a very simple technique to characterize the bacteria morphologically. Okay, because every bacteria look different. So we use this one just to differentiate the bacterial colonies. It's a very useful tool for morphotyping, right? Otherwise most of the colonies look alike on plate, so we decorate the bacteria by adding this chemical. Okay, that's what. Otherwise by default Pseudomonas fluorescens is not red in color. Okay, don't carry the message like that. We artificially, we all do some makeup like that we did for bacteria. Okay, so bacteria looks beautiful now, isn't it? So you can also do this, right? So any other colonies? Okay.
Only that first. This is our first group.
Yeah.
Others, second is pure. Nothing is there. Here some bacteria.
If nothing is there, I really appreciate them, and you could do the microbiology experiment without contamination. So that's really beautiful. Yeah.
This is the third group.
Yeah, unfortunately the training is getting over on Monday, so if any one of you want to follow it up, you are most welcome. If you feel any one of the colony is suspected to be a Pseudomonas and you suspect them to be a transformed one, please come back to us. We will give you the primers, the forward and reverse primer. You can use it, and the insert is there, then that is the transformed colony. Okay. Now you, but some plates I am seeing fungi, so that means it's a very poor handling, right? So always remember, this kind of work you have to give a lot of time. You cannot do in a hurry. If you do in hurry, it is bound to fail. In microbiology, these two are two opposite poles. Okay, you have to give enough time, and you need to have a lot of patience and planning. 99% planning only, right? You prepare everything well in advance. Okay. All microbiology and pathologists, what we do most of the time, we prepare food for microorganisms. It's big work all the time, correct? So never ever streak your plate the day of your experiments, right? 24 hours before you prepare your plates, all right? And keep your plates in a setup like this. Keep it at room temperature, right? Next day you come and see you just weed out the plates which is throwing some contaminants, right? Then take the best plates and take them on board. If it is antibiotic amended medium, you straight away put it in cold room. You can use it for a week maximum. Okay, you have to reduce the workload on a daily basis. So if you prepare around 30-40 plates, right? So it's a one-day job, right? Similar work, coordinated effort. So it is easy to prepare 2 liters media and 1 liter media with the same effort. You understand? So prepare the plate and pour the plate, then keep it in your lab, right? I do not know how many of you noticed in my group, we don't store media in flask. Did anybody noticed? You will not see a flask with media in my team. I would say it is completely banned in my group. I don't like to see that, right? So why? Any idea? Remelting, you have to again melt it. Again, melting means what? The nutrient composition will be different. Chemical composition is bound to change. With repeated heating and using, you are giving it's not the media what you are giving the way it is expected or it is found in the literature. Okay. So the pipeline is, you prepare media in the morning, from autoclave, you come to your lab and it is poured right away. Okay. If you are not able to come to lab, At the time of the media coming out of your autoclave, we have provisions in the lab. You can keep the media at a molten state at least for 3-4 hours. It never solidifies. Okay? So maybe after your classes, you come back in the evening, maybe around 5:30 or 6 o'clock, and the media is already in the molten state in a setup. We have a kind of heat block, and or sometimes we put it in the incubator at a high degree, around 60 degrees Celsius. You pour it, then you go. The advantage is plates are available for your work at least for another 3-4 days. Every day you don't have to prepare this media in your lab. Okay, you try to do this, and I'm sure you will enjoy doing more and more work. Okay, and if you think, okay, every day I have to make media, people used to rush into the labs. Most of the time they come around 10:30, 11 o'clock is the cutoff time, and in a hurry you make your food, then you are bound to spoil it. Right now, so don't do that. So 11 o'clock cutoff means you come by 8:30 and start making your media, right? And send it for autoclave, then you take a break. Okay, so some kind of mindset you have to change. Otherwise, we are really spoiling this kind of experiments if you really start making media on a daily basis. That too after 10:30, right? That is not allowed. I do not know about microbiology how the system works, but here we have a cutoff. 11 o'clock is autoclaving time. Yes, it's centralized autoclaving. It's a beautiful system which is working in our division now. Okay, so with that we will formally conclude this today's practical.